Good evening, everyone. I'm Dr. Janice from Biochemistry for Dr.Polaris, and this is a recall session for biochemistry. Now, you might be wondering why this recall session is meant for and what's the benefit of this. Now, the main purpose of the recall session is to reinforce whatever you have gone through the videos and to make sure that you're able to recollect some and probably even clear some of your doubts. Now, before I begin with the recall session as such, I just remembered that there's this very interesting concept called as a cone of learning, which was given by Edgar Dane. He is an American educator. So he came up with this cone of learning. Practically all of us have different styles of learning. Some of us like to, just read. Some of us like to draw. Some of us like to watch YouTube videos and so on. So there are different methods. But how much of that can we recollect is what matters in the end. So as per the cone of learning, okay, it is said that whatever be the method of learning, if you go and just check yourself after 2 weeks and try to recollect whatever you have learned, it is said that we remember 10% of what we read, maybe in the form of textbooks. You read your textbooks and all. So we remember only 10% at the end of 2 weeks of what we read here. And we remember 20% of what we hear. Now, what do you mean by that? Now, some of us like to hear podcasts and all that stuff. So probably, 20% of whatever we hear through podcasts, we can recollect. 30% of what we see. Now, some of us like to, see, pictures, diagrams, cycles and all those things in a nice graphical format. And some of us can learn faster that way. So we can remember 30% of what we see that way, okay, at the end of 2 weeks. Now, 50% of what we see and hear is what we are able to recollect. Now, this could be in the form of your videos. Okay, now whatever you see and hear, you're able to recollect only 50% of that, if that's your learning strategy. Then 70% of what we say and write. So today's recall session, I expect all of you all to, interact and, come up with answers. I'll be showing you all a couple of MCQs. I want you all to interact, tell me what are the possible right answers, tell me why those, some of the other options are incorrect. So we can have a very interactive session that way. So hopefully you're able to recollect whatever you learned from this session after 2 weeks. At least 70% of it can be remembered. And the last higher level, you might think, what is a higher level? That would be 90% of what we do. Now, that would happen only at the patient bedside when we apply certain concepts, in the clinical side, or probably, work in the simulation lab and so on. So when we do things, we tend to learn much more. Okay. And when we recall like this, we also tend to learn quite a lot. So without wasting any time, we'll begin with the first, the recall session with the first topic, that is glycolysis. Are you all ready, everyone? Yes, hopefully all of you all are ready. So I'll just help you all out with this, as in I'll be showing you all a set of questions. Now, some of the questions may have more than just one right answer. So you will have to identify those right answers and then, probably put it up in the chat box here. You can see there's a chat box. So you can just put it up there and let me know what the right answer is. And then maybe I might give you all a chance to, explain, or, we can just have a normal discussion regarding those points. So that was about how we'll be going about with today's session. So we'll just start off with the first topic. We'll begin off with the first topic, that is glycolysis. Are you all ready? Yes? Okay. So the first question is for glycolysis. Glycolysis occurs in all cells. Occurs in the cytoplasm, which is a subcellular site. The starting material is glucose. Pyruvate is the end product in the absence of oxygen. And E, lactate, is the end product in the presence of oxygen. What do you think the right answer would be? Now, remember, there can be multiple right answers. So I want you all to just go for it. You can write down your right answers in the chat box. What do you think the right answer would be? Yes, I want some kind of interaction here. See, there can be multiple right answers. So you can just go about, or whatever, if you just think, one of them might be the right answer, you can just write just one option, correct option. Yes, Shritam has come up with option A. Very good. Yes, that's the right answer. Anything else about glycolysis? Yes, glycolysis occurs in all cells, practically all cells. Yeah, you can go wrong, as Dr. Aishwarya rightly put it. Yeah, you can go wrong. This is the right place to make mistakes. So if you go wrong, we are here to correct you. Don't worry about saying the wrong answer. It's all fine with us. So just go ahead and put in your right options. Yes, just one answer, is it? Option A. What about option B and option C, option D and option E? Yes. Shritam, would you like to add something more other than option A? Because as far as I know, there are more than just one option for this question. Yes, can we unmute Shritam?
Ma'am, it could be C.
Yes, it could be C. Did you say C?
No. Yes, ma'am.
Yes, yeah, it could be C, isn't it? So glycolysis involves the oxidation of glucose. Yeah, C would be right. And then, yes, we have Natasha here saying B and C are also right. Okay. Yes. So we'll just have a look. I think we have Shritam and Natasha here. If you combine both the answers, they're all right. So we'll just go ahead and see what the right answers here. Glycolysis, as you all know, is the oxidation of glucose. It occurs practically in all cells, and the subcellular site is cytoplasm. The starting material would be glucose, yes, but the end product would be different depending on whether or not oxygen is present. So in the presence of oxygen, the main end product is, yes, anyone would like to have a go at it? What would be the end product at the end of, glycolysis happening in the presence of oxygen?
Ma'am, in presence of oxygen, it could give pyruvate, and in absence
Yes.
of oxygen, it could get.
True. Correct. So that's right. So the incorrect answers here are D and A, B, and C are the correct answers. Remember, pyruvate is the end product in the presence of oxygen, whereas lactate is the end product in the absence of oxygen. So I hope you all have got this concept right. Moving on to the next question. The next question is, the key enzymes of glycolysis: A, glyceraldehyde 3-phosphate dehydrogenase; B, hexokinase or glucokinase; C, enolase; D, phosphofructokinase; E, phosphoglycerate kinase; and F, pyruvate kinase. Yes, what do you think the right answer would be? You can enter your correct answers in the chat box. Anyone? Yes, go ahead and put in your answers. See, you might get it wrong or right. It depends if you have gone through the topic. But this is a nice time. I mean, this is a good opportunity for you all to get some of your concepts cleared also, and to know what the right answer is. So Shritam over here has said, except E, all are correct. Okay. Anyone else would like to answer? Yes? D. Okay, someone has said, Swadesna has said the option is D. Phosphofructokinase is a key enzyme. Asta has said D and F. Okay. Anyone else? Okay, Shibani has come up with the option B. Okay. So now you need to remember this. There are three key enzymes for glycolysis. Okay, there are three key enzymes of glycolysis. So anyone, could you just narrow down your options or come up with more options and then tell me what are the key enzymes of glycolysis?
B, D, and F.
B, D, and F. Very good. Who's this?
Astha.
Asta. Hi, Asta. Yes, you're right. B, D, and F are the right answers. They are the key enzymes of glycolysis. Asta, can you tell me what does B do? What's the function of B?
It converts glucose to glucose 6-phosphate, and it is an irreversible step.
Very good. So hexokinase or glucokinase is the first key enzyme, and it helps to convert glucose to glucose 6-phosphate. The second answer that she had mentioned is option D, which is for phosphofructokinase. And phosphofructokinase helps to convert. Asta, would you like to do the honors?
Yes, ma'am. Fructose 6-phosphate to fructose 1,6-biphosphate.
It's not bi, okay? It's bis. Okay, just remember, bisphosphate. So phosphofructokinase converts fructose 6-phosphate to fructose 1,6-bisphosphate. And finally, we have the last key enzyme, which is pyruvate kinase, and that helps to convert
phosphoenol pyruvate to pyruvate, which is
very
the
good. Yes, very good. So pyruvate kinase helps to convert phosphoenol pyruvate to pyruvate. So I hope you all have got this right. That remember, there are three key enzymes. You have hexokinase or glucokinase, which is the first key enzyme. The second one is phosphofructokinase, and the third one is pyruvate kinase. Now, what do you mean by a key enzyme? A key enzyme is one which helps to regulate the entire pathway. So depending on the activity of these enzymes, if the activity is quite reasonable and high, then the pathway will operate. If the activity is low or totally negligible, then the pathway will not operate. So that's the meaning of key enzyme in simple terms. The next question, moving on to the next question, third question. Energetics of aerobic glycolysis. Remember, aerobic glycolysis is glycolysis which occurs in the presence of oxygen. So hexokinase utilizes 1 ATP. As you can see, there's a negative sign here. The negative sign here indicates utilization of ATP. The second one is phosphofructokinase, which utilizes 1 ATP. The third one is glyceraldehyde 3-phosphate dehydrogenase enzyme, which produces 4 ATP. That is 2 molecules of the substrate gives rise to 2 ATP. And then phosphoglycerate kinase gives rise to 2 ATP again. And then we have pyruvate kinase, which gives rise to 2 ATP. Now, which among these are the correct answers? Yes, we need to have more contributors to the answers. I can see only few people just contributing the answers. You can go ahead and just, unmute yourself if you think you know the right answer, but it would be better if you just write in the right option first in the chat box so that we can ask you all to unmute. So Sritam over here has come up with the option A and B. He thinks A and B are the correct options. Anyone else? Yes, there are more than 2 right options for this question. So can we add some more right options to this? C and E. Okay. C and E. Patasa has said C and E. Shritam has added E now. Okay, he has added A, B, and E. Anyone else? Asta has said A, B, and E. Okay, both Shritam and Asta are, stuck with the same answers. Yes. Anything else? Anyone else would like to contribute? We have a lot of participants here, and, it would be nice if everyone puts in the right answers and not just, see what's happening. Please enter your right answers in the chat box. Yes. Even D. Okay, Asta has said even D is the right answer. Well, Asta, you're right. Very good. Asta, can you tell me why is C not the right answer? You can unmute yourself.
Yes, ma'am. Actually, 2 into 2.5 ATP should be the correct answer. So it would yield to 5 ATP.
Correct. Very good. Very well done, Asta. That's absolutely right. So all the options here that you can see here, energetics of aerobic glycolysis is brought about by, yeah, the option A is correct. Option B is right. Option D is right. And option E is right. Now, C would be incorrect. The reason for this is because if you just have a look at the step, which is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, you get 2 molecules of NADH. So that NADH actually is utilized, and 1 molecule of NADH produces 2.5 ATP. So 2 molecules of NADH would produce 5 ATP and not 4 ATP as it is shown in the option. So the option C would be incorrect in this case. So moving on, I hope you'll have got this right. Okay, I hope you'll have got this right. So remember, A, B, D, and E are the right answers. Moving on to the next question. The 4th question for glycolysis. Hemolytic anemia can be due to A, sorry about this. Yeah, A, pyruvate kinase deficiency, B, hexokinase deficiency, C, phosphotriose isomerase deficiency, and D, enolase deficiency. So hemolytic anemia, as you know, is because of destruction of the RBCs. So hemolysis takes place. Why would hemolysis takes place? That we'll discuss later. But for now, which of the enzyme deficiency of glycolysis would lead to hemolytic anemia? You can enter your right options in the chat box. Okay, Sheetam has said it's A. Anyone else would like to add? Yes, come on, we can have some more answers. Anyone else? Now, remember, this question has just more than one right option. So you can keep on adding whatever you think is the right option. Okay, Fatima said B. The option is B. Okay. Any more answers? Yes. I think we can unmute Fatima or Sheetam. Fatima or Sheetam, we can unmute them. I think Sheetam has already got a chance to talk earlier. So we can move on to Fatima. Fatima can, give her views why she thinks option B is right in this case. Oh no, Fatima left. I hope she did not get scared because I asked her. Yes. Anyone else? Okay, so we have Sheetam over here who has said the option A is correct. And we also have Fatima who said that option B is correct. Now, let me put this across. Remember that, if there's deficiency of any of the glycolytic enzymes, it will obviously lead to hemolytic anemia. The reason is very simple. Okay, you just remember that, RBCs, mature RBCs, as such lack mitochondria. So if the mitochondria is not present, okay, they lack mitochondria. So if mitochondria is not present, what's going to happen is they're not going to have enough of energy to carry out, their various metabolic processes. So energy has to be obtained from some source at least. So main source of energy production in case of RBCs is anaerobic glycolysis, okay, to put it straight. So if there is any kind of enzyme deficiency in the pathway, okay, of glycolysis, any kind, right from the beginning till the end, you have seen so many enzymes. If any of those are deficient, it will lead to hemolytic anemia because the RBCs, will require some energy, and that energy is only obtained through glycolysis. So out of all these enzyme deficiencies that you can see A, B, C, and D. Any of these enzymes, if they are deficient, can lead to hemolytic anemia. So all the options are right. Now, the most frequent cause for hemolytic anemia among the glycolytic enzyme deficiency is caused due to pyruvate kinase deficiency. That's the most common reason for hemolytic anemia. When you just go and have a look at, the incidence of or the prevalence of these deficiencies that exist. So among the enzymes, pyruvate kinase deficiency is the most common one. So we move on to the next one, the next question. The fifth question, conversion of pyruvate to acetyl-CoA, A, occurs in mitochondria. B, the energetics is 3.5 ATP per molecule of pyruvate. C, it's an oxidative decarboxylation reaction. D is an irreversible step. And E, formation of acetyl-CoA, which enters into citric acid cycle. Now, which among these options are right? You can enter your right options in the chat box. Yes, can we have some answers here? Asa has said yes, D. She thinks D is the right answer. The conversion of pyruvate acetyl to acetyl-CoA is an irreversible step. Okay, good. Any other answers? This question has more than one right answer. So you can put in your right options. Okay, we have Shibani over here who has come up with 4 options. Let's see what are her 4 options. Shibani has come up with options A, C, D, and E. She thinks A, C, D, and E are the correct options for this question. Very good. Shibani, can you unmute yourself? Shibani, are you there? Can you please unmute yourself? I would just like to know, why do you think B, option B is not the right answer? Shibani? Okay, actually, Shibani has come up with the right answer, but I can't get her to unmute herself to answer this question. Okay. Can anyone tell me, or can anyone tell me why is what Shibani has said, options A, C, D, and E are correct for this question? Can anyone tell me why is option B incorrect? You can unmute yourself. Yes, Asta has said that. Yes. Asta, can you unmute yourself?
Yes, ma'am. Conversion of
yes.
one molecule of pyruvate will lead to 3.5 ATP generation, and if we take two pyruvates, it will lead to generation of 5 ATP.
Okay. What did you say again? Can you just repeat it once again?
Generation of total pyruvate to acetyl coenzyme A will lead to formation of 5 ATP.
Yeah, why 5 ATP?
We take 2 molecules of pyruvate because
Okay.
that starts from glycolysis, which in DHAP gets converted to glycerol, glyceraldehyde 3-phosphate. So 2 molecules of pyruvate are formed in glycolysis, and we have to consider both the molecules.
Okay, I'll just ask you one question because this is concerning the conversion of pyruvate to acetyl-CoA. Okay, this the enzyme that is involved is pyruvate dehydrogenase complex. Now, in that complex, if you just go and see, what is the main coenzyme which is formed in the end, or what is the reducing equivalent which is formed in the end of the chain of reactions that take place? Can you remember? What is formed in the end? So you have a lot of conversions that take place when pyruvate is converted to acetyl-CoA. The enzyme is pyruvate dehydrogenase complex. It's a multi-enzyme complex. So you need a couple of coenzymes and different more than one enzyme. And you have some reducing equivalent which is formed in the end. Can you tell me what that is?
NADH.
Yeah, NADH is formed. And how much of ATP does NADH produce?
1.5.
2.5, isn't it? 2.5. 2.5. So this option, that is B over here, 3.5 is incorrect because it is actually 2.5 of ATP that is generated because of the formation of NADH when pyruvate is converted to acetyl-CoA. So that is per molecule of pyruvate will form 2.5 ATP because of the production of NADH in the end. If you're talking about how much of pyruvate, like you have, how much of energy is produced by this particular reaction after glucose is oxidized. Once glucose forms pyruvate, you know, there are 2 molecules of pyruvate that is formed from glucose. So in that case, the total energy that is produced from 2 molecules of pyruvate would be 2 into 2.5. But here the statement that is seen here is, the statement is very simple. It says that 3.5 ATP is generated per molecule of pyruvate. That is an incorrect answer because it is 2.5. You have NADH, which is produced as a reducing equivalent. And you know, when that enters into ETC or the electron transport chain, it generates only 2.5, not 3.5 ATP. So that was about the options. So among all these options, the option B is incorrect. Now, what do you mean by oxidative decarboxylation? Anyone? What do you mean by oxidative decarboxylation? I hope you'll have understood why it is 3.5 ATP. It is not 3.5, why option B is incorrect. In case, yes, Shibani has said carbon dioxide is released in oxidative decarboxylation. So you see that whole term, okay, oxidative decarboxylation is what really happens here in this particular reaction that is catalyzed by pyruvate dehydrogenase complex. You see there are 2 parts of the term. You have oxidation and decarboxylation. So there is removal of hydrogen, and at the same time there is removal of carbon dioxide. So these are the 2 processes that take place when pyruvate is converted to acetyl OA. So moving on, I think we have completed the questions for glycolysis. I hope you all found this very helpful, and we have been, you know, successful in trying to cover up all the essential parts of glycolysis. So we move on to the next topic, which is citric acid cycle. I hope you all are all ready for this. So the first question here is, the subcellular site of citric acid cycle is A, mitochondria, B, cytosol, C, endoplasmic reticulum, and D, nucleus. What do you think would be the right answer in this case? Shibani has said it's option A. Yes, Shibani, very good. Very well done. It is option A, and I think most of you all have no confusion about this. It's very good that you'll all know. Asta and Natasha, all of you all have said option A. Yes, option A is the right answer. So the subcellular site where citric acid cycle takes place is the mitochondria, and all the enzymes required for citric acid cycle exist within the mitochondria. That's why the site is mitochondria. So A is the right answer. Now moving on to the next question. In the energetics of glucose oxidation, A, glycolysis produces 7 ATP. Pyruvate dehydrogenase complex produces 5 ATP. TCA cycle produces 10 into 2 ATP, which is 20 ATP. B, 1 mole of glucose in aerobic condition forms 32 ATP. And finally, option E, 1 mole of glucose under anaerobic condition forms 30 ATP. Which among these are correct options? You can enter your correct options in the chat box. Remember, this question has more than just 1 correct answers. So Asa has come up. That was really quick. If this was fastest fingers first of KBC, you would have got it. You would have entered into KBC, Kaun Banega Crorepati. So she has come up with A, B, C, and D, which are the correct options for this question. Asa, can you please unmute yourself and tell me why is E not the right option?
Total ATP produced under anaerobic condition is 2 ATP, not 30 ATP.
Very good, very good. Yes, true. So under anaerobic conditions, you have to remember when 1 mole of glucose is not oxidized, as in it's converted in the absence of oxygen, you get 2 ATPs and not 30 ATP. So option A, B, C, and D are right in the energetics of glucose oxidation. Moving on to the next question. TCA cycle is amphibolic, B, anaplerotic, C, inhibited by fluoroacetate, arsenite, and malonate, and D, occurs in certain cells. Can you please enter the right options for this question? This question has more than one right answers. Yes. I don't see anyone entering the right answer for this. Yes, Asta has come up with A and C. And Kirti Raj, please correct me if I pronounce your name wrong. He has come up with A, B, and C as the right answer. So, Kirti Raj, can you please unmute yourself? Yes. Why do you think D is not the right option? Asta said it's also B. Okay, very good. Kirti Raj, can you just tell me why is D not the right option? Okay, Soumya Patel also has said the answer is A, B, and C. Very good. I see most of you all telling me the right answer, but I also need some kind of interaction with you all, explaining why the other option is not right. Yes, Kirtiraj, very good. You said it right. It occurs in all cells, not in certain cells. TCA cycle doesn't occur in certain cells. It occurs practically in all cells. So we'll just go through each of the options one by one. So as we know, TCA cycle is a very important cycle. It's the common oxidative pathway for the metabolism of all, carbohydrate, protein, and fats. So TCA cycle is amphibolic. Can anyone tell me what do you mean by this term amphibolic? You can unmute yourself and then let me know what it is. Yes, Asa has said you have both catabolic and anabolic pathways. So you have, substances that are formed from the intermediates of the pathway, and you have certain substances which can generate certain intermediates of the pathway. So I will just give you some examples of your catabolic pathways in TCA. Catabolic pathways would be, your utilization of alpha ketoglutarate, which is one of your intermediates of TCA cycle, where you can form a glutamate from it, okay, through transamination reactions and so on. So alpha ketoglutarate can form glutamate, and then again glutamate can form, certain important substances in the body. Likewise, you have also malate, which can form glucose and so on. So you have catabolic role over here. And then you also have anabolic, where you can form, sorry, I just messed up the entire thing. I'm so sorry about that. The anabolic part would be, your alpha ketoglutarate would be formation of glutamate. And then from glutamate, you can get other non-essential amino acids, and even glutamate can go on to form, certain, contribute to the formation of purine and pyrimidine ring structure. So that is formation, anabolic. Catabolic would be the breakdown of certain amino acids or fats to form the intermediates of the pathway. So that would be in case of, like, say, certain amino acids like phenylalanine, tyrosine, and so on. When they are broken down, they form fumarate, which again is one of the intermediates of the TCA cycle. So that way, that's the meaning of amphibolic, where you have both things happening at the same time, your anabolic as well as catabolic. Now, the next thing, what do you mean by anaplerotic? Anaplerotic, anyone? Anaplerotic? Okay. So, yes, somebody has said, Asta, yeah, reaction that can fill up the intermediates are anaplerotic. Very good, Asta. So, yes. So, as you know, certain intermediates, if they are utilized for the formation of other important products, okay, in the body, over a period of time, the concentration of these intermediates, you have your alpha ketoglutarate and, succinyl-CoA, malate, all those things, the concentration will gradually drop. And if the concentration of these intermediates of the TCA cycle drops, what happens is the TCA cycle will not operate. So it's very important to replenish all these intermediates through the help of anaplerotic reactions. Can anyone give me one example of an anaplerotic reaction that takes place to fill up the intermediates? Anyone? Any one reaction? You have a couple of reaction. So I can tell you one example where, one of the intermediates, like say, for example, your succinyl-CoA, for instance. So yes, somebody has said here, pyruvate carboxylase. Okay. So you have different types of anaplerotic. That would not be, that is a part of your, okay, yeah. Okay, that would be also. So you have different types of reactions that can come and fill in. You have transamination reactions, which can fill up your alpha ketoglutarate, then you have oxaloacetate. So all those reactions are anaplerotic. Then the second option, that is the third option, inhibited by fluoroacetate, arsenite, and malonate is right in case of TCA cycle. Fluoroacetate inhibits aconitase, which is an enzyme which converts citrate to isocitrate. Arsenite is again another inhibitor of the TCA cycle, which inhibits the enzyme alpha ketoglutarate dehydrogenase, which converts alpha ketoglutarate to succinyl-CoA. And finally, malonate, which is an inhibitor of the enzyme succinate dehydrogenase, which converts succinate to fumarate. So these are the three main inhibitors of the TCA cycle. So the last option here occurs in certain cells is incorrect. It occurs in all cells. So the correct answer for this question is A, B, and C. The next one is TCA cycle is regulated by A, citrate synthase, B, isocitrate dehydrogenase, C, alpha ketoglutarate dehydrogenase, D, hexokinase, and E, enolase. What are the steps or points in the TCA cycle which are regulated? Can you please enter the right option? Yes, Chandani has said the option is A, B, and C, and even Shibani. I think Shibani has got a chance earlier to mention why the answers are right or wrong. So we'll move on to Chandani. Can Chandani unmute yourself and tell me why D and E are not right? Or Shibani, can you unmute yourself and mention why D and E are not correct in this case? Yes? Yeah, correct. Chani, you're right. So the options here for question number 4 here, A, B, and C, these are the enzymes of the TCA cycle, whereas D and E, hexokinase and enolase, are enzymes of glycolysis. So definitely those are not the right answers. Moving on to the next question. Choose the correct order. The first here that you can see, acetyl-CoA, citrate, alpha ketoglutarate. Then you have succinyl-CoA, malate, fumarate, and oxaloacetate. Then option B, you have these intermediates. Option C, you have another set of intermediates, and D here. So find out, can you just tell me what is the correct order of the intermediates of the TCA cycle? Which among these options are right? Shibani is very quick. She has come up with option A. Anyone else? Yes. Okay, most of you all have come up with the right answer saying that option A is correct. Yes, option A is correct. Just remember, these are the intermediates. That is of option A. Now moving on, I think we're done with TCA cycle, and we can just have a look at the next option. The next topic, that is regulation of blood glucose. So the first question that comes under regulation of blood glucose, the normal postprandial blood glucose level is A, 70 to 100 milligram per deciliter, B, 100 to 140 milligram per deciliter, C, 70 to 140 milligram per deciliter, and D, 120 to 140 milligram per deciliter. What is the right answer? Asa has said the option is A. Anyone else? Shibani said it's A. Remember, this is postprandial blood glucose. There's a difference. Okay, it's not, there are 3 types of, blood collection when you want to estimate blood glucose. Okay, Chandni has said it's option B. And even Asta now has changed option to option B. Shibani has changed to B. Yes, B is the right answer. Chandni, can you just tell me what do you mean by postprandial? What do you mean by postprandial? So there's 3 ways by which you can collect, yes, after meal. Chandni said after meal. Immediately after meal, or is there a particular time period after meal that you need to collect the blood sample for glucose estimation? Generally, it is after 2 hours post your meal. So 2 hours after a meal, that is considered as a postprandial sample. When you collect the blood sample from a patient or from an individual, 2 hours after he or she has taken his last meal, we call it as a postprandial. So in that case, the normal levels will be around 100 to 140 milligram per deciliter. You also have fasting blood glucose, and the normal level would be 70 to 100, where a person fasts for 8 to 12 hours. Then you have a random blood glucose level, that is irrespective of whether the individual has taken his meal or not. You take the blood sample and the level will always be in the range of, blood glucose level will be in the range of 70 to 140 for random. Now moving on to the next question. Causes of hyperglycemia are all except A, diabetes mellitus, type 1, type 2, and gestational diabetes. B, hypothyroidism. C, Cushing's disease, acromegaly. D, metabolic syndrome. E, pancreatic diseases. F, drug-induced or steroids. Drug-induced, maybe by the use of steroids or beta blockers. What are the incorrect options? So here you need to find out what is the incorrect options. All of these will cause hyperglycemia except, which is the incorrect one? Which one will not cause hyperglycemia? Anyone? Okay, Natasha has said the option is B. Natasha, can you just unmute yourself and tell me why is B the right? Why is B the answer here in this case, in this question?
Ma'am, since metabolism is low, BMR will be low because thyroid hormone is less.
Okay. And what do you know about thyroid hormones? Are thyroid hormones known to be hyperglycemic or hypoglycemic in nature?
They're hyperglycemic.
Hyperglycemic. Okay, very good. So whenever there is any decreased level of T3 or T4 that would ultimately result in hypothyroidism, what happens is the glucose level also tends to decrease. So this would, option B would be an incorrect answer. Okay, hypothyroidism would not cause hyperglycemia. Rather, it would cause hypoglycemia. Now, the other options, I'll just go through the other options as well. See, all these, that is option A, C, D, E, and F are correct for the question, that is causes of hyperglycemia. Only option B is incorrect. So this is the only one which does not cause hyperglycemia. So in case of diabetes, you know, there is always hyperglycemia. Either because of insulin deficiency or insulin resistance, there can be an increase in blood glucose level. The next one that you can see, option C, Cushing's disease. Can anyone tell me what is Cushing's disease due to? Or what is predominantly increased? Which hormone is increased in Cushing's disease? Cortisol, Chandni. Yes, Chandni, correct. Cortisol is a hyperglycemic hormone. So cortisol again can increase the blood glucose level, leading to hyperglycemia. The next option that you can see is acromegaly. And which hormone is generally elevated in acromegaly? Growth hormone, Asta, very good. So growth hormone again is a hyperglycemic hormone. And again, if there's excessive growth hormone, that can lead to hyperglycemia again. The next thing that you can see is metabolic syndrome. So this is a cluster of conditions. You have multiple things happening in metabolic syndrome. You can find abdominal obesity where there is increase in the waist circumference. There will be hypertension, increase in blood pressure. There can be impaired fasting glucose, okay, that can lead to hyperglycemia. And also there'll be increased level of triglycerides and decreased levels of high density lipoprotein. So these are all the different, things or criteria to confirm the diagnosis of metabolic syndrome. So hyperglycemia is one of the criteria in metabolic syndrome. The next thing that you can see is pancreatic diseases. Again, the pancreas is known to produce insulin and glucagon. So if there is a imbalance in the ratio of insulin and glucagon, again, there can be hyperglycemia. And finally, drug induced. You can see there are two drugs mentioned here. One is steroids and the other is beta blockers. Now, steroids, as such is glucocorticoids, and glucocorticoids again has a hyperglycemic action. Again, it can lead to hyperglycemia. Beta blockers, on the other hand, this is a class of drugs that is used especially in the treatment of heart failure and atrial fibrillation and so on. So these beta blockers basically they prevent the release of insulin, and that can lead again to hyperglycememia. So all the options here can lead to hyperglycemia except hypothyroidism. Moving on to the next question, that is question number 3. Common symptoms of hypoglycemia are. Now, I think most of us would know what the symptoms are. Each of us would have faced this at some point of time in our life. So the option A, the first option is headache. Second option is anxiety and confusion. The third option is sweating. D, anuria, and E, seizures. Which among these are the common symptoms of hypoglycemia? Now, this question has more than one right answers. So you can enter all your answers in the chat box. Hypoglycemia is a condition when the blood glucose falls below 60 milligram per deciliter. So except D, Asa said except D. Very good. That's right. So except D, anuria. What is anuria? Anuria basically is when there is no urine output, or there is minimal urine output of less than 100 ml per day. So anuria generally is not a symptom of hypoglycemia. The rest are, as you can see in the options here, headache, yes, you get that. Anxiety, confusion, yes. You also sweat, and yes, seizures can also happen, especially when the blood glucose falls very, very low, that is below 40 milligram per deciliter. So the common symptoms of hypoglycemia are A, B, C, and E. Moving on to the next question, question number 4. Hyperglycemic hormones are all except A, glucagon, B, insulin, C, epinephrine or adrenaline, D, glucocorticoids, E, adrenocorticotropic hormone, and F, growth hormone, and then G, thyroxine. What is the one which is incorrect? Yes, Asta has come up with the option B, and Charni as well, B. Shibani, why do you think it's A? Okay, Shibani has immediately changed her option to option B. So I see most of you all are right. So remember, you have only one hypoglycemic hormone and you have several hyperglycemic hormones. So your hyperglycemic hormones would be A, glucagon, C, epinephrine, D, glucocorticoids, E, adrenocorticotropic hormone, F, growth hormone, and G, thyroxine. Insulin is the only odd one out which has an hypoglycemic effect. That is, it decreases the blood glucose level. The next question, glucagon inhibits A, glycogenesis, B, glycolysis, C, HMP shunt, D, lipolysis, E, TCA cycle, and F, gluconeogenesis. Now, I know most of you all might get stuck here looking at all these pathways and cycles, but just get this one thing right, okay? What is glucagon? Glucagon is a hyperglycemic hormone, okay? So it should stimulate all the pathways which lead to increase in blood glucose and inhibit all the pathways which decrease the blood glucose level. So what would that be? Asa has come up with A and E. Okay, that's right. She has added D also. So right now Asa is saying that A, E, and D are the right options. Okay. Okay. Anyone else would like to add? Only Asa has replied to this. Remember, glucagon should stimulate all the pathways which increase blood glucose level. okay, breakdown of certain substances where glucose is obtained in the end. And then glucagon should inhibit the pathways which decreases the blood glucose level. Yeah, Asa has added more now. B also she has added. Okay, she thinks it's A, B, and D. Okay, now we'll quickly just go through each and everything. Okay, I'll just go through each of those pathway. We'll start off with this. As I said, glucagon, the main aim is to increase the blood glucose level. Now let's see glycogenesis. What happens here? In glycogenesis, glucose is utilized to form glycogen. So obviously glucagon should inhibit this pathway. So the first answer is correct. Glucagon inhibits glycogenesis. The next one, glucagon and glycolysis. Just see this one. Glycolysis again utilizes glucose to give you something. Okay, glucose is utilized. So again, glucagon should inhibit glycolysis. So B is also the right option here. HMP shunt again also involves a utilization of glucose. It's an oxidative process. So again, glucagon should inhibit this pathway. Now I'll just leave this lipolysis towards the end. TCA again is utilization of glucose in the form of acetyl-CoA. So again, glucagon should inhibit this pathway. And then F, gluconeogenesis, that is formation of new glucose. Okay, formation of new glucose from other sources. So glucagon should stimulate this pathway so that more and more glucose is formed, and that can lead to elevation in blood glucose. So obviously this last option is not correct. So we are left with only A, B, C, D, and E. Now we'll see lipolysis. Okay, why can't lipolysis be the right or wrong answer? We'll see that. So lipolysis, you remember, happens in the adipose tissue. Can anyone tell me which is the main enzyme involved in the breakdown of stored fat in the adipose tissue? What's the enzyme called? Can anyone tell me? You have a specific enzyme which is very sensitive to certain hormones, and I've already told you the clue. Yes, Kirtiraj has come up with that. So hormone sensitive lipase is the hormone which is present in the adipose tissue and helps in lipolysis, that is breakdown of stored fat. Now this hormone sensitive lipase is stimulated by glucagon and inhibited by insulin. So again, lipolysis can't be the right option in this question. So the correct options for glucagon inhibiting are A, glycogenesis, B, glycolysis, C, HMP shunt, and E, TCA cycle. D and F are incorrect answers. So that was it about regulation of blood glucose. We'll move on to the next topic, which is ammonia disposal and urea cycle. So quickly moving on to the first question, transamination is A, transfer of an amino group from an amino acid to a keto acid. B, transfer of an amino group from a keto acid to an amino acid. C, regulated by transaminases. D, irreversible reaction. And then E, all amino acids participate in transamination reaction. What is the correct option? Now this question has more than one right answer. Asa has said the first option A, yeah, that's the definition of transamination. You're right. What's the second right? Yes, very good. So the second right answer is regulated by transaminases, which was given by Asta and Kirtiraj. Now together they have said the options A and C are yes, very good. So transamination reactions involve the transfer of an amino group from an amino acid to a keto acid, so that in the end you get a new keto acid and a new amino acid. And yes, the enzymes that help to catalyze this entire reaction are known as transaminases. And this is a reversible step. And one more thing, the last option, can you see all amino acids participate in transamination reaction, which is false. So can you tell me what are the exceptions of which amino acids cannot participate in transamination reaction? Can you tell me which are the amino acids which cannot participate in transamination reactions? What are the exceptions? There are 3 amino acids that way. Any idea? Okay. The 3 amino acids which do not participate in transamination reaction are lysine, threonine, and proline. These are the 3 exceptions. Rest of the other amino acids will usually participate in transamination. Moving on to the next question. Significance of transamination is that it produces non-essential amino acid. B, concentrates nitrogen in glutamate. C, releases ammonia. D, energy production occurs with excess of amino acids. And then E, glycolysis and lipolysis also occurs as a result of transamination reaction. What would be the right option? This question has more than one correct answer. Asta Shah has come up with A, C, and D. Okay, Kirtiraj has added B. Okay. So Asta and Kirtiraj, if you combine both your answers together, that would be the right option. So the correct options here are A, B, C, and D. E, obviously, glycolysis and lipolysis has nothing to do with transamination reaction. But the other things, as you can see, formation of non-essential amino acids is possible with transamination reaction. Then you also can have nitrogen, which gets concentrated in glutamate. Along with that, ammonia can also be released, okay, through certain reactions in the transamination. And even energy production can occur with excess of amino acids. So all this is possible with transamination. The next question is, oxidative deamination involves the removal of ammonia and oxidation. B, the major site includes liver and kidneys. C, provides ammonia for urea synthesis. D, occurs via glutamate dehydrogenase and amino acid oxidase. And E, deamination of histidine occurs. What is true about oxidative deamination? This question has more than one right answer. Can you enter your right answers? Yes, Asta has come up with B. Kirtiraj has come up with A, B, C, and D. Very good, Kirtiraj. That's right, A, B, C, and D. Can you unmute yourself? And can you tell me why is E the incorrect answer? Or you can write down your answer in the chat box also. No issues. Why do you think E is the incorrect answer? So for the rest of you all, I'll just tell you all, oxidative deamination is, yes, a reaction which involves both oxidation and removal of ammonia, which occurs through deamination. So both these processes take place, and the major sites include liver and kidneys. And the major thing at the end of oxidative deamination is to form ammonia. And that ammonia is utilized for the formation of urea. Now, this occurs with the help of 2 enzymes, glutamate dehydrogenase, which converts glutamate to alpha-ketoglutarate. And amino acid oxidase, you have 2 types. You have L-amino acid oxidase and you have D-amino acid oxidase. Yes, Asta has come up with the answer why E is not the right answer. So she says that histidine undergoes non-oxidative deamination, which is true. Okay. So remember, deamination can be of 2 types, oxidative and non-oxidative. So one form of non-oxidative deamination is the deamination of histidine, which does not involve oxidation. Very well done, Kirtiraj and Asta, for giving the right answer. So we move on to the next question. Classification based on disposal of ammonia include A, ammoniotelic, that is ammonia is water. B, uricotelic, that is ammonia is converted to uric acid. C, ureotelic, that is ammonia is converted to urea. D, humans are uricotelic, and E, snakes are ammoniotelic. Which among these are the right answers? This question again has more than one right answer. Shibani has quickly come up. Asa has also come up. So we'll see Shibani's first, A, B, and C. Asa has said that all 3 are correct. Or sorry, all 5 are correct. Kirtiraj again has come up with the 3 answers. Now Asa has changed it a little bit and she has said that D is not the right. So we'll go on with, I'll tell you who's the right person who has come up with the right answer. I think it is Shibani. Shibani has come up first with the right answer. And she said that A, B, and C are correct, whereas D and E are incorrect. Remember, humans are not uricotelic. They do not secrete uric acid. What do they secrete? Urea. Okay. So definitely D is the incorrect option. And snakes again are not ammoniatelic. Again, they secrete uric acid. Okay. That's the end product, the excretory product. So uric acid. So snakes, reptiles as such, they are uricotelic. The rest of the other options are correct. A, B, and C are correct. Moving on to the next question. Clinical features of ammonia toxicity include slurring of speech, B, tardive dyskinesia, C, chorea, D, blurring of vision, E, hepatic coma, and then 7, mental retardation. Which among these are the clinical features of ammonia toxicity? This question has more than 1 correct options. Yes, Kirtiraj has come up with A, C, and F, I think, and Asta A, D, E, and F. Very good. Asa, very good, done, very well done. Oh, Kirtiraj said it's D. Okay. So Asa actually is the first person who has come up with the correct answer. It's A, D, E, and F. Okay, so these are the common features of ammonia toxicity. Now, why is B and C not correct? Okay, I'll just tell you what is B. B is tardive dyskinesia. That is, it's an involuntary repetitive movement of the face. Like, usually such type of people will have frequent grimacing. That is, a kind of a facial, kind of a facial movement where the person looks like he's always frowning or he's always in pain. He doesn't really want to do it, but it just so happens that his face looks like or appears like that. So that's called as grimacing. And another thing is there'll be frequent eye blinking. So these are commonly seen in tardive dyskinesia, and frequently this tardive dyskinesia occurs especially after a patient is on some long-term psychiatric medication. So that's about tardive dyskinesia. It doesn't really have to do anything with ammonia toxicity. The other one is chorea, and chorea basically is again involuntary movements of the legs, arms, and face, and so on. So here again, the reason for this, it's actually a jerky, uncontrollable movement that is seen in case of chorea. And here the main reason is, the reason for chorea is due to elevated levels of dopamine. So again, that could not be one of the clinical features of ammonia toxicity. So the correct options in this case would be option A, D, E, and F. So with that, we are done with the questions for ammonia, disposal, and we'll move on to the next one, that is translation. Yes. So the first question in translation, the requirements of translation include A, you need mRNA, B, you need tRNA, you need ribosomes, you need energy in the form of ATP or GTP, you need enzymes and certain factors, and finally you need F, ribosomal RNA. So which among these are the requirements of translation? Yes. Are you all tired? Is it? After such a long session? I can't see any options at all. Yes, anyone? Yes, we have Kirtiraj over here who says that A, B, C, and D are right. And then we have Shibani also, A, C, E, and F. And then we have Asa, a lot of mixed reactions I can see over here. Some say this is required, that is not required. Okay. And then Asa has added one new. So again, we have Asa. Asa, you're doing really well. You have this one, I think you're getting most of the answers right. So Asa, again, she's right here in this case. So all these are the requirements of translation. You need mRNA, which contains the codons, and based on the codons, you have the amino acid sequence, added to the new protein chain, polypeptide chain that is synthesized at the end of translation. You need tRNA. tRNA helps to bring or carry the amino acid to the site of protein synthesis. You need ribosomes. So you have two types of ribosomes. You have 80S ribosomes in eukaryotes and 70S ribosomes in prokaryotes. This is the factory house in which proteins are synthesized. You need energy. That is also true. And you also need enzymes like amino acyl tRNA synthetase and factors such as initiation factors, elongation factors, and releasing factors. The only incorrect answer here is ribosomal RNA, which is not necessary for translation. Moving on to the next one. The stages of translation include A, activation of amino acid, B, initiation, C, elongation, D, termination, E, post-translational modification, and F, splicing of RNA. Which among these are the stages of translation? This question has more than one right answer. Except F, all. So ask again here, he has mentioned that except F, everything else is the right answer. Very good. So yes, translation is a synthesis of proteins from mRNA. So the first step that it involves is the activation of amino acid. The second step includes initiation. Third step includes elongation. Fourth step includes termination of the protein synthesis. And then finally, post-translational modification to make sure that the protein that is synthesized is fully matured. There are various types of modifications that occur. And lastly, you can see F, that is splicing of RNA. Now, splicing of RNA, especially messenger RNA, is an example of a post-transcriptional modification. It doesn't occur in translation at all. So that's totally different. So all these are correct except option F. Very good, Asta. We'll move on to the next question. So initiation in translation includes A, dissociation of 80S ribosome into 40S and 60S. B, formation of binary and ternary complex. C, formation of 43S pre-initiation complex. D, activation of mRNA. E, formation of 48S initiation complex. F, formation of 80S initiation complex. And then G, termination. I think this should be easy, I guess. And yes, again, this question has more than one correct option. So Asa has said the correct option is A, C, and E, is it? Yes, others, you can put in your right option. She's added F also. Anyone else? Okay, so I think, yes, A is wrong. A is wrong, is it? Okay. Okay, A is wrong. Okay, that's what Shibani has said. Anyone else? What happened to the others? We have a lot of attendees I can see here. So I would expect others also to pitch in the right answers. Yes, I will tell you the right answer. Okay, so initiation, as you know, is the initiation of protein synthesis, and there are several steps that come under that. So the first step is A, dissociation of 80S ribosome into 60 and 40S. B is also, it also happens. So you have both binary and ternary complexes taking place. You have certain initiation factors also which come into play here. Then formation of 43S pre-initiation complex. This is when the ternary complex binds to the 40S subunit. Then you have activation of mRNA. This happens especially when ATP, and then you have this, the cap added to mRNA and all that. So again, that is also an essential criteria of initiation. Then formation of 48S initiation complex also takes place. Here the 43S pre-initiation complex and the mRNA fuses together to form 48S. So that is also an important step. And then finally F, formation of 80S. This is where both your 48S and your 60S combined together, and there is some release of certain factors. So all these steps are important and are a part of initiation except termination. So the last option G is incorrect. The rest of the options are right. Moving on to the next question. Translation in eukaryotes. A, ribosome is 80S, where you have a larger subunit 60S and a smaller subunit 40S. The initiator RNA is methionine tRNA. Start signal is initiator codon, okay, that is AUG of mRNA, and is preceded by methyl GTP. B, initiation factor is initiation factor 1, 2, and 3. Termination factors are releasing factor 1, 2, and 3. So these are all the options. Can you tell me which one is correct? Okay, these are all the options. You have to tell me what is correct among these options from A to E. In case of eukaryotes, remember this is in case of eukaryotes, not prokaryotes. And this question has more than one right answer. So can you just put in your right options? Asa has come up with A, B, and C. Okay. Asa, can you unmute yourself and can you tell me why D and E are not the right options? What you have told is absolutely right. A, B, and C are the right options here. Now, why are D and E the incorrect options?
Ma'am, I think that initiation factors, we have our eukaryotic initiation factors, that is ELF. Sorry, EIF.
Yeah, EIF, yes.
Yeah. And I am not sure about termination factor.
Yeah. So just remember that in case of eukaryotes, okay, all your initiation factors or termination factors or even elongation factors for that matter, it always, gets preceded by the letter E, small e. So small e stands for eukaryote. So that's how you differentiate between the factors for prokaryotes and eukaryotes. In case of prokaryotes, you just have initiation factor, whatever number, how many numbers are there. But in case of eukaryotes, you have, eukaryotic initiation factor, eukaryotic initiation factor 1A, 3, and so on. There are so many initiation factors for eukaryotes, not just 3 that is mentioned here. 3 initiation factors is just for prokaryotes. For eukaryotes, you have plenty. Okay. Likewise, the same holds true for termination factor. You have that eukaryotic, small e preceding before the releasing factor. So the rest of the others option that you can see, yeah. Asa is terminating is by stop codon. Yeah, terminating is by stop codon. Plus, you need certain factors also. Okay, you need certain factors also to help to release all, the protein, the mRNA, and all those things in the end. So yes, termination occurs by stop codons, but you need other releasing factors also to, unassemble the entire thing, like mRNA, tRNA, the protein chain, all those things happen in the end. So that was it about question number 4. So remember, A, B, and C are the right answers here. Now, I'll just ask something about prokaryotes. What is a ribosome in prokaryotes? If it had to be translation in prokaryotes, what would be the ribosome be? That would be 70S. Okay. Yes. And then, yeah, Kirtiraj, that's true. What you have said is true. Correct. 70S. Yes, Asta. And what would be the initiator RNA in case of prokaryotes? It's not methionine tRNA. It is formyl tRNA. Okay, formyl methionine tRNA. Yes, Kirtiraj got it right. Formyl methionine tRNA is the initiator RNA present in prokaryotes. So that is one thing you need to remember. Moving on to the next set of questions for translation. Various types of post-translational modifications are A, protein folding, B, modification of amino and carboxy terminals, C, loss of signal sequences, D, proteolytic degradation, E, modification of amino acids, and F, deletion of prosthetic groups. What are the various types of post-translational modification? This question has more than one right answer. Yes? Post-translation modification are a set of modifications that happen once the immature protein is formed at the end of translation. So there are specific groups added or specific modifications that occur in the structure so that you get a fully functional protein. So Asa has said A and C. Okay. Anyone else would like to add? Okay, she has added D. Anyone else? Okay, I think people, yes, all. Okay, Kirtiraj has written all and then with a smiley, upside down smiley. Well, it's not all. Okay, it's not all, but one of them is incorrect. So now can you just modify your answers and tell me? One of them is incorrect. Which one is the incorrect one? D? No. Okay. Anyone else would like to give a wild guess? E, Asa said E, no. Okay. Okay, now I'll just tell you. Okay. First, we'll start off with protein folding. Yes, it is a post-translation modification. You need certain proteins called as chaperones, which help in, making sure that the proteins get a three-dimensional form. Modification of amino and carboxy terminals. Yes, that can also happen in post-translational modification, where you can have phosphate groups added or, carboxy groups added, phosphorylation, carboxylation, methylation. All those modifications can occur in case of the amino and carboxy terminal. Loss of signal sequences. This also can happen. This is a very important step, especially in protein targeting, where the protein has to be told where it's supposed to go after it is synthesized in ribosomes. The protein cannot exist within the ribosome there. I mean, it has to go to the site where it's supposed to travel to, or where it's supposed to remain, maybe in the mitochondria or the cytoplasm or so on. So who really tells the protein to move from the ribosome to the cytoplasm or the mitochondria? That happens with the help of signal sequence. So once the protein reaches its site or its destination where it's supposed to be present within the cell, the signal sequence have to be cut off or trimmed, or there should be loss of signal sequence. So again, that's an example of a post-translational modification. Proteolytic degradation again is an example where extra amino acids can be trimmed or removed. That is also again a post-translational modification. Modification of amino acids again can happen, but the last option you can see, deletion of prosthetic groups. That's an incorrect answer. Addition of prosthetic groups usually happens in post-translational modification. So I'll give an example of acetyl-CoA carboxylase. That's an enzyme which is required for fatty acid synthesis. Now, the prosthetic group that is present in acetyl-CoA carboxylase is biotin. So the addition of biotin happens in the last step of the post-translational modification of acetyl-CoA carboxylase. Likewise, we have even cytochrome C, again as a protein. So again, the prosthetic group is added there, and the prosthetic group added in cytochrome C is usually heme. So addition of prosthetic group is a step of post-translation, not deletion. Moving on to the next one, inhibitors of translation are A, streptomycin, B, tetracycline, C, chloramphenicol, D, erythromycin. What do you think would be the right answer? A, B, C, and D. Wow. Okay, Asa has said all four are right. Which is right? True. All are correct. They are inhibitors. Remember, these are all antibiotics that are used in the treatment of various bacterial infection. So streptomycin and tetracycline basically bind to the 30S subunit, okay, especially in prokaryotes. So 30S subunit is a smaller subunit and prevents elongation, whereas chloramphenicol and erythromycin again are inhibitors of prokaryotic translation, and it binds to the 50S subunit, the larger subunit. So all of these are inhibitors of translation. Now moving on to the last one, that is genetic code, and I think this should be quite easy. So the first question of genetic code, all are features of genetic code. A, universality, B, specificity, C, degeneracy, D, overlapping, and E, commoness. What are the features of the genetic code? So except one, everything else is the right answer. Can anyone tell me? Yeah, except C. Kirtiraj has said except C. Okay, he has changed it to D. Yes, Kirtiraj. Very good. Kirtiraj, can you tell me why is it not D? Very good. So yes, what Kirtiraj has just, yeah, it's non-overlapping. Very good, Kirtiraj. So genetic codon code, as you know, is a collection of codons. You have a sequence of 3 nitrogenous base arranged like that. So first and the most important thing is that genetic code is universal. Okay, the same codon, the same will code for the specific amino acid in all species. Like you have UUU, which codes for phenylalanine in humans, bats, cats, whatever, you just name it. So it's universal. Second one is the genetic code is very specific. That means one codon will code for only one amino acid. That is, if you take UUU, a codon, it will code only for phenylalanine and not tyrosine. So it's very specific. So one codon codes for one amino acid. Degeneracy means, it is said that there are around 64 codons, out of which 3 are you have 61 codons which actually help in, protein synthesis as such. 3 of those are your stop codons. So 61 codons are there for 20 amino acids, 20 standard amino acids that are normally incorporated within the protein chain. So it is said that as per the degeneracy rule, that one amino acid can be coded by more than one codon. Okay. So it's something like, like me, for instance, I'm an individual and I have, if you just take the example of an ID proof, I can have more than one ID proof. I can have a passport, I can have a voter's ID, I can have a ration card, I can have different kinds of ID proof. But all this ID proof, if you just take individually, all this ID proof, it will show or indicate that it belongs to me. Likewise, the same thing holds true for genetic code. So one amino acid can have multiple codons, okay, just like your ID proofs, okay. But if you take one codon, it always codes specifically for one amino acid. So it's something like that, okay. So that was about degeneracy, and yes, it is non-overlapping and it is comma-less also. Now we move on to the next one, degeneracy of genetic code. Some amino acids are coded by more than one codons, reduces harmful effects of mutation. C, permits alternate base pairing of anticodon in tRNA and codon in mRNA. D, incision or deletion of base sequence can change the reading frame. And then E, codons are read continuously as 3 bases without any punctuation. Which of these are correct about degeneracy? This question has more than one right answer. A and B. Okay, very good. There's one more. Would you like to add? There are 3 right options. A and B is right. Yes, some amino acids I just explained. So some amino acids are coded by more than one codons and reduces a harmful effect of mutation. That's the whole purpose of degeneracy. And then the last one, E. E is not correct. That doesn't really explain degeneracy, insertion and deletion. Okay. So the correct options are C also. Kirtiraj has just added C also. So Kirtiraj is right. A, B, and C are the right answers. So degeneracy explains all these 3 options, whereas D and E doesn't really explain what degeneracy is all about. It's nothing to do with degeneracy of genetic code. Moving on to the next question. Consequences of frame shift mutation are all except A, coding for a different amino acid, B, generation of terminating codon, C, premature termination of protein synthesis, D, initiation of translation. Which among these will not be the consequence of frame shift mutation? Yes, Asa, absolutely right. Initiation of translation, yes, that's not going to be a consequence of frame shift mutation, because frame shift mutation can lead to the formation of a stop codon, and that can lead to termination of translation, not initiation. So that is one thing that you have to remember. So in this case, the correct answer is A, B, and C. And what doesn't really happen is D, initiation of translation. Moving on to the next one. Genetic code contains dash base sequences: A, 2, B, 3, C, 4, D, 5. Very good, Asta. You have not given time for anyone else to answer. Anyone else would like to add? Yes, it is B. 3 base sequences are usually present in a set of codons. So very good. That is the right answer. And I think we are done with our presentation and the MCQs. I think we had a lovely time. I guess I might have, went on beyond my 1 hour time limit. I hope everyone enjoyed, and we can have, we can have more topics and, more discussions like these. I hope you all did not get bored, and hope you all are able to at least, recollect something at least from whatever has been told in this recall session. If you all have any doubts or anything, you can always post it up right now in the chat, or you can unmute yourself, or there is a question and answer tab here in Zoom. You can open that and you can put in your questions if you have any doubts. I hope all of you all enjoyed this session. If you all have any doubts, you can just send in a message right now itself, or we can wrap up this session. Dr. Aishwarya, what do we do?
Hi, Dr. Janice. It was such an informative session. I'm so happy that I could listen and get back to, being a first-year student. I'm so happy to listen to you. And I hope even our dear students are also happy. So guys, it's just a recall session. Make sure that all the points that ma'am has discussed today is being taken down, and you just put down these points in your exam papers, and that's what the tutors or the faculty or your teachers would be looking for in your papers. So that's the exact points which she discussed today, and I'm so happy that she took it in a very interactive way as well. So would like to know if you have any questions. If there are no more questions, and we can wind up the session, and would like to know how the session was. Can you just drop a message in the chat box how the session was, how was ma'am's class today, so that both of us, she gets to know a feedback.
True.
Right, ma'am?
Yes, that's true.
Just drop in a message, all of you, please.
Yes.
So that we get to know. And in the meanwhile, we do have upcoming sessions of recalls like this for the other subjects. And even one more session from Dr. Janice also would be there. And it will be like on the 21st of this month, if I'm not wrong. But definitely we're letting you know a detailed schedule, and we have 4 other topics as well. So do wait and do tune into Dr. Polaris sessions. Yes, ma'am. They are all happy with your session. Fabulous session. Thank you so much. It was amazing.
Thank you, everyone. I hope you all had a nice time. I hope this was quite informative as well, and not too much, bogging you down with so much of information. But I hope it helped you out at least to some extent. And yes, we'll be having another session on the 21st, hopefully. And we'll have another set of topics where we can clear out some more doubts and, have such interactive sessions, question and answer, recall sessions. So hope to see you all next time on 21st.
Ma'am, someone is asking how to contact you, ma'am.
I guess that would be possible only through Dr. Polaris, right?
Yes, you can let us know if there is anything. Ma'am is so sweet. Definitely, if you have any queries or anything which has to be posted to any of the faculties of Dr. Polaris, do let us know in the chat bot which is available, or even the community managers whom you are in contact with, or even the campus ambassadors whom you are in contact with. They will direct it to us. You can directly contact with the respected faculties. Don't worry on that. All our faculties will be available for you guys, because that is what we are here for, to help you guys complete the whole mission of going through this journey. So don't worry, we will always be available, and keep tuned for new sessions. And until then, it's bye from both of us.
Yes.
Okay.
And happy Valentine's Day.
Yes,
what a day, right?
I really love the way you clear our concepts. Really love the session. Okay, thank you so much, guys. It's so overwhelming to listen to your comments and see them. I think ma'am is equally, in the same way like how I feel. So again, happy Valentine's Day to all of
Yeah.
you.
Yeah.
I hope your Valentine's Day dinner has happened already, or
Yeah, there's
it's
still time. It's already, I mean, it's just 8.
yes. Let them go have a good dinner, ma'am. All the best,
Yes.
guys.
Yeah,
Keep
all the
tuned.
best.
All the best. Bye. See you. Good
Bye-bye.
night.
Good night.
Thank you,
See you.
ma'am.
Thank you. Thank you so much.